Hello, my name is Tanisha Williams, and I'm the Burpee postdoc in botany at Bucknell University. I'm presenting this work with my collaborators Jeff Heim and Dr. Chris Martin, also from Bucknell, and with Chloe McGuire, Amy Robleski, and Dr. Rebecca Bleege Bird from the Pennsylvania State University. Today I'm going to share how we are using genomics methods to understand how the Martu people have influenced the distribution of a staple food source known as wamula throughout the Western Australian desert. Western Australia is home to the only biodiversity hotspot found on this island continent and has plenty of desert habitat. Within these harsh conditions, there is a great diversity of plants and animals. The desert is also home to Aboriginal peoples as well. One such group is called the Martu people. The Martu consists of several Aboriginal peoples that have family connections and similar dialects and cultural traits from the Great Sandy, Little Sandy, Gibson, and Nullarbor deserts. Because of their lifestyle, the Martu people have extensive traditional ecological knowledge. The Martu people have a tradition of burning and hunting for resources for their community's well-being, and they also do this to increase resource availability, promote diversity, and population stability. Our research group has strong ties with the Martu community, and it builds upon over 20 years of research with the Martu. Most recently, work has been done to understand how the Martu promote the distribution of Solanum species through seed dispersal, and we have found that plants are found closer to communities and occupation sites. We know that humans have had a significant role in crop and animal domestication. There's a lot of documentation on the evolution of agriculture for most regions of the world. But what about species that are not domesticated but still used as a staple food source? With the Martu people, we are trying to assess the Martu's influence on the distribution, genetic diversity, and dispersal patterns of a non-domesticated staple food source known as Solanum diversiflorum and locally known as wamula. Wamula is a spreading prickly shrub and often found in sand. This is a tasty and easily harvested and processed food staple. The first two pictures above show wamula's flowers and fruit. The last picture shows women and children at a wamula processing site. Due to the pandemic, we were not able to go to Australia, but we were able to use the resources that we had and started working with the 154 samples of wamula. Previous studies have shown that there is some correlation between seed dispersal and distance to Martu sites, so we wanted to see if the movement of wamula shows up in its DNA. We assessed the genetic diversity and structure of populations close to and managed by the Martu people compared to populations outside of the Martu tidal lands. So let me orient you to this map. The red area shows the Martu tidal lands. The darker gray areas show other Aboriginal tidal lands. Our DNA falls into 4 clusters. Within those red Martu tidal lands, those are the Martu samples. And then we have the Great Northern Highway group, the Wolf Creek group, and the Buddhist Station group. So we extracted DNA using a FastPep kit, used a GBS approach. All assemblies were done in iPirad, and population genomics analyses were done in R. So overall, results show that wamula does not have a lot of genetic diversity or population differentiation. Inbreeding is low for this group. Results show a clear separation between the Martu populations and from the wild populations. We see that wild populations are more isolated from one another. They have lower genetic diversity and have higher levels of inbreeding compared to the Martu populations. These results confirm what we have seen in a preliminary study and what we expect is true. The Martu are maintaining genetic diversity within populations in their tidal lands. The movement of plants between populations is helping keep inbreeding low when compared to wild population and assisting gene flow and connectivity of these populations. As we can see here in this structure results, the wild populations are in blue and Martu populations cluster in these green and red unit. Zooming into the Martu tidal lands populations, we can see these 2 distinct genetic units, and they break down into known movement corridors between language and family groups. So the activities of Indigenous people have profound impacts on species distributions, and we are excited to understand more about how these relationships can give us new insights into anthropological relationships, plant ecology, and in conservation. Again, we want to thank the Martu community for this wonderful collaboration. We also want to thank our funding sources, and we are currently on our way to Australia to learn more from the Martu and to increase our sampling among those wild populations. So stay tuned for more next year. Thank you so much for coming to my talk, and I look forward to speaking with you in the future about this amazing research. Thank you.